everyone. What an amazing day. Yeah, this is first time for me in Assam. So I heard a lot about Assam and the amount of stuff you guys are doing, including Giri. He's an amazing doctor, saving a lot of lives. We see his emails, messages all the time. And then Jayditya, Nanishwar, everyone is taking a lot of initiative here. And I knew Assam government is ahead of other governments in India. But today I'm seeing action. A minister is agreeing to do so many actions on the stage itself. I've never seen in any government meeting a minister is agreeing to do the things immediately on the stage itself when everything is being recorded. So you guys are doing an amazing job. Many congratulations for everyone, and please continue to do this as well. You guys will act as a role model for the rest of India. So first of all, I would like to thank the Biodiversity Board and the government of Assam and Jayditya and Yaneshwar for giving me this wonderful opportunity to come over to Assam for the first time, even though I've been trying to come here for the last 3 years. So Yaneshwar asked me to talk about the values of venom or applications of venom. And I just added one more word here, unknown values of the venoms. I normally call venom as a biotreasure. Okay? So even though in the country we are talking about the big four, as everyone else mentioned, in northeast India, you got so many varieties of snakes. And most of these venoms have never been characterized properly. So think about the entire world. So we got over 600 species of venomous snakes and over 200,000, that's 2 lakh species of invertebrate species. Okay, including tarantulas, true spiders, scorpions, centipedes, everything. We got far too many species across the world, and less than 1% of these species have been researched properly in science. Okay, so remaining venoms have never ever been touched in science. So this is a biotreasure. The more you hunt, the more you're going to find out. That's what we normally call. Okay, so venom holds potential to treat almost every single human disease. So that's why venom research is not just to develop or improve antivenom. It's mainly to find out so many other cures for other diseases. So including us, scientists and clinicians, many people don't know the full potential of the venom, and that's why I call this as an unknown values of venoms. So someone previously mentioned about the difference between venoms and the poisons. So venoms, as you may know, it's a modified form of saliva. It's secreted by modified form of salivary glands called venomous glands. Okay? So when a tissue is specialized in secreting or synthesizing and secreting a material, and it's got a specific organ to deliver that material into the host tissues, that's called venomous animal. Okay? Whatever the material is secreted and delivered is called venom. It's a simple. And if you take poison, it will be secreted from any tissues, and normally it will not have a system to deliver this poison into animals. So when you ingest or when you eat those animals or plants, if you die, then it's a poisonous. Okay? But here, venom must be injected into the animals. So usually venomous animals like snakes, scorpions, tarantulas, true spiders, these animals produce venom mainly to capture their prey. Because we are not their prey, so they are not producing venom to capture us or eat us. But unfortunately, they can use the venom for different purposes. But if you look at poisonous animals or poisonous plants, they mainly secrete poisons in order to defend. So venom composition. So what is in the venom? So we know in venom, nearly 95% of the venoms are proteins, okay, and peptides. So if you look at the snake venoms in the top here, it's a viper venoms. It's got proteins at a different molecular weights. You can find low molecular weight proteins, high molecular weight proteins, and it's got enzymes and also non-enzymatic proteins. And each one is doing a different job. By doing so, they are trying to arrest the physiological functions of prey as quickly as possible. And when the venom is injected into our body, it's trying to do the same thing. Okay, so elapid venoms, if you look at like a cobra venoms, they will have a lot of small molecular weight proteins, very few high molecular weight proteins. But if you look at viper venoms, they will have proteins at different molecular weights across the spectrum. And similarly, if you look at the scorpions and tarantulas, they will have tons and tons of peptides, tiny little peptides, even like 10 amino acid size peptides. very few high molecular weight proteins. Okay, and imagine thousands of peptides doing different jobs. What will happen to their prey or the animals which they are biting? So that's why we need to try to understand what each one of these components is doing in the venom. Why these animals are spending their energy trying to produce these venom molecules in order to capture their prey. And these venoms can change. I mean, people have talked about venom variations. So venom variation is huge across within the species. Okay, if you see a snake, for example, Russell's viper here, and then if you get a venom from Tamil Nadu, the venom will be varying. Within the species, there will be a huge variation based on different factors. And that's why we need to study more and more about the venoms. And then because of the wide varieties of venom proteins and the peptides present in the system, obviously they will do a lot of different functions. For example, if you look at the viper snakes, they predominantly attack our hemostatic system or circulatory system. Okay, they will either induce random blood clots or they may cause a random bleeding. And they can also cause huge amount of tissue damage around the bite site. And if you don't take treatment properly, that will lead to permanent disabilities. Because if you are coming to a hospital very late, we have to go for amputation. That's another problem. And if you look at the elapid snakes like the cobras, they predominantly attack our nervous system and try to cause paralysis. Okay, so these proteins or peptides have precise targets in the body. Okay, and they are purposely produced to induce specific effects in their prey or in host animals. And they do have only minimal off-target effects. That's the beauty of these venoms. Okay, compared to any other drugs available in the market or plant-based compounds which may bind to more than one target. In some cases, like plant compounds can bind to thousands of targets in our body, but these molecules are likely to bind to precise target. That's the beauty of this. So for example, if you look at this is our blood clotting system, and all these blue ovals are venom proteins, venom serine proteases, and these serine proteases can specifically act on specific clotting factors or specific receptors on platelets in order to either cause procoagulant effects, which means they will make blood clots in our body, or anticoagulant effects, which means they will prevent the normal blood clotting in order to induce excessive bleeding. So they have precise target. So as we all know, we are using venom to produce antivenom. Antivenom is the only treatment. I wouldn't call only treatment. It's an essential treatment for snake bites because we should also give other medications or secondary medications to treat snake bite cases. So antivenoms are produced by collecting venoms of regional importance. So for example, in Assam, people are thinking about this new 6 medically important snakes. So you can collect the venoms from these snakes, mix them all together, inject into large animal models, and then you can collect the blood from those animals and purify the IgG, and then finally you can provide Fab fragment or Fab2 fragment. That's how antivenoms are being produced at the moment. So in order to produce high quality antivenom, we need a high quality venom. In order to get a high quality venom, we need a high quality healthy animals. Okay, if you get a diseased animal or already damaged, you are not going to get a high quality venom. If you don't get a high quality venom, you are not going to get high quality antivenom. So these things are really important. And we are also talking about the next generation antivenoms. The antivenom technology we are using at the moment, it's around 150 years old. Okay, the technology hasn't been improved quite a lot, and it has got a lot of side effects. Okay, antivenoms can cause allergic reactions, not in every single patient, but in some patients it can cause allergic reactions. Okay, and it's also expensive because they are coming from animal origin. Luckily in our country, the government is providing antivenom for free of cost. But in many countries, people need to pay for antivenom. So it's got its own merits and at the same time demerits. So many people are working in order to develop alternative therapeutics, including small molecule inhibitors for venoms and recombinant antibodies, including nanobodies. But all these research, we need a high quality venom. Similarly, we also need diagnostic kits because we don't have any diagnostic kit in the country, in most of the world, except in Australia. So if you have a diagnostic kit in a rural area, immediately we can test some patients using their blood or urine and confirm the presence of venom in their body. In the next generation of kit, we can also confirm which type of snake bit them. Okay, so as soon as you confirm the presence of venom, immediately you can administer few vials of antivenom before you refer them to tertiary care hospitals. For all these research, we need venom. Without the venom, we cannot do any of this research. So that's also important. And then venoms, as I mentioned right at the beginning, it's playing a critical role in drug discovery. Okay, in the past, people were developing drugs based on the plant materials because plants got millions of materials. But now many people, and including pharma companies, turn towards venoms because, as I said, there are over 200,000 invertebrate venomous species available. Over 600 venomous snakes are available, and most of these venoms have never been characterized for a drug discovery. And as you can see, all these drugs are already available in the market, and they are all derived from venom molecules. And there are so many other drugs are in pipeline in different stages of clinical trials. So the more you hunt, the more you're going to find out, because these venom molecules are target specific, so they can exert specific effects, and they may not cause any side effects, or they may cause only minimal side effects, and most importantly, they are untapped resources in drug discovery, so we can use them very nicely. Venoms can also be used in basic science, and across the world, so many people are doing basic science work. Okay, as I said at the beginning, we need to really understand what is in the venom. Okay, and what is it doing? Then you can understand what sort of functional effects they will have in patients or in prey. Okay, then that can translate into developing new diagnostics or therapeutics for snake bites and also for other diseases. So we can also do evolutionary studies to understand why these proteins are being produced and their relationship with our proteins, mammalian proteins. And we can also understand the basic composition and the different peptides, venom variations, all these things you can identify. Okay, by doing this, you can improve the knowledge about venoms, and you can reveal their scientific and clinical potential, and this will also promote conservation of these venomous animals. Once you understand the potential values of these animals, you're going to protect ultimately, and the public can also protect these animals. Then venoms in education. In Tamil Nadu, we've been thinking about including snakes and snake bites in school and the college curriculum for some time. While we were discussing about including theoretical materials, we also thought about some practical classes. As you may know, in schools, in higher education, particularly in +1, +2, and in most of the biological courses, life science programs, BSc Biochemistry, Microbiology, whatever it is, we do a lot of practical classes in our course. For example, simple protein quantification. We normally use BSA for protein quantification. Why can't we use some venom? Okay? Because you use BSA, you remember that throughout your life. Your protein quantification very in the first year of your BSc Biochemistry or Zoology, whatever it is you might be doing, you're going to remember that. Similarly, we can do a lot of immunoassays. ELISA, antivenom is available. It's a cheap, we can buy in this country, and we can get venom. We can do simple ELISA. Enzymatic assays, we normally take salivary amylase assay. We take saliva and quantify the amylase activity. Why don't we quantify serine protease activity or metalloprotease activity or phospholipase A2 activity from venom? By doing these type of things in your education, you can learn more about venoms. By learning more about venom, we can develop awareness about snakes and snake bites in our community. Okay, if you are studying something about venom in your college, you can go and educate your family members, relatives, friends about the venoms, their dangers, and also their biological values. Okay, so we can also realize the value of venom, and this will also promote curiosity-driven research in future generations. The more people come to know more about venoms will significantly improve the amount of research we are doing in this field in future, and this will help developing better diagnostic and therapeutic for snake bites, and it will also improve the drug discovery research, and it will ultimately promote the conservation of venomous animals. And then you see in news quite often about venoms in illegal trade. Okay, people all the time say, oh, if a kite come and bite in my tongue, I'll get euphoria, or I'll become high. Okay, and the customs officials are seizing venom all the time in airports and railway stations. All these things are mainly happening because of the lack of knowledge about venom. So everyone should really know what is venom and what it does. Okay, once if we know the dangers of venom, then people are not going to use this type of things. And the customs officials or any other officials who are seizing these venoms, they should also have awareness about venoms and how they look like and what sort of simple tests they can do in order to confirm whether it's a venom or not. Because sometimes people say, oh, I got 1 kilogram of scorpion venom. Just imagine the number of years it will take to generate, to collect that much of venom. So we need to really understand whether it's a venom or something else. It's really important. And once if we develop that knowledge, basic knowledge about venoms, we can easily control the illegal trafficking of venoms. So what are the challenges in using venoms? Okay, it's easy to say we can use venoms for everything, but there are thousands of challenges associated in using venoms. First thing, collecting venom is a challenging task. As you all know, snake rescuers, we need well-trained experts in order to collect venom without damaging the animal. Okay, and at the same time, you need to get a high quality of venom. And in many cases, like in scorpion, tarantulas, or true spiders, the yield of venom, the amount of venom you are collecting will be very low. So there will be a scarcity for venoms all the time. And there are lots and lots of rare species. Like Dr. HT mentioned this morning, there is quite a lot of rare species available here, particularly in this part of India. And collecting these venoms is a real challenge. Okay, we can't easily get that. And then venom variations. That's causing a lot of difficulties in maintaining snakes in captivity and collecting venom for research. And the cost of venom, based on the quantity of venom you are getting from animal, the cost will vary, and not every researcher can afford to buy the venom. And then prohibition of exports for international use. Many countries, including India, they banned the export of animals and animal products. Even though the legal aspect says that for life-saving research this can be allowed, but it doesn't happen always. And everyone should understand that in 2010, Nagoya Protocol was formed. And so many countries across the world, I think 145 countries signed this Nagoya Protocol. This protocol protects the biodiversity of each and every country. You can easily share venom or any plant material, any materials from India with any countries across the world. Whatever the research they do, you will get the benefit share. Okay, that's what this Nagoya Protocol, it protects biodiversity of every single region and every single country, and therefore the government should step forward in order to share the venoms with other countries in order to promote better research everywhere as well. And then lack of investment in venom research. Until recently, like only in the last 10 years, snake bites have been started talking again quite a lot, and many funding agencies are coming forward and funding this research. But in the past, people were suffering to get funding for venom research because again, due to lack of awareness. So we need to join hands, as Romulus Whittaker mentioned, we all need to join hands. So in this room, lot of experts, lot of different stakeholders available. Snakebite research cannot be done just by an individual. Like a scientist cannot do everything what they want just by themselves. Clinicians cannot do everything what they want but just by themselves. Only by joining hands we can do a lot of things. By doing a lot of things, we can improve venom research, we can try to get better medications and diagnostics for snake bites, and we can also try to develop better medications for other diseases, and most importantly, we can try to protect the venomous animals in order to protect our biodiversity. If you are interested, we publish an article called The Value of Venom in Royal Society of Biology. The journal magazine is called The Biologist. You can look at this as well. And I would like to thank all our collaborators and the funding agencies. And finally, I would like to thank the government of Assam for giving me this wonderful opportunity again. Thank you.